Hi everyone, and welcome to today's lecture on amino acid metabolism. As always, I just want to remind you that there will be the chalkboard notes following each slide for you to pause the video and jot down your notes. Now, when we talk about amino acids and proteins, a critical part of their molecular structure is that they contain nitrogen. And nitrogen is very valuable to us and to plants and bacteria. So we have to have ways to then isolate or get that nitrogen from amino acids and from various sources to then use them, and then to get nitrogen to be able to make amino acids and proteins. Because remember, metabolism is both, it's building and breaking things down. So here we have two terms that are important ultimately to amino acid metabolism. We have nitrogen fixation and we have assimilation. Now, the first one, nitrogen fixation, is the reduction of nitrogen gas to ammonia, so to NH3 or NH4 plus. And it also includes the oxidation of nitrogen gas to nitrites and nitrates. So those are NO2 minus and NO3 minus. And when it comes to nitrogen fixation, the three main sources that I want you to be aware of are bacteria, the Haber process, which is the industrial way of us doing this process, and then there is also lightning. Now, the other term, assimilation, nitrogen assimilation, that's basically when ammonia is directly incorporated into amino acids. And that's done for us by plants and bacteria using an enzyme called glutamine synthetase enzyme. So now, a moment ago, I mentioned nitrogen fixation, and I said three ways to get that process are bacteria, the Haber process, and lightning. And so on this slide, when we say three forms of fixation and we say biological, industrial, atmospheric, biological is the bacteria-based form of nitrogen fixation. That's when they're using that nitrogenase enzyme, they're breaking the triple bond of nitrogen gas, and they're basically doing a six-step ATP dependent process for fixation for us. The industrial method is then represented over here. That's the Haber process that I mentioned a moment ago. And basically, what you see happening here is nitrogen, 1 part nitrogen, is mixed with 3 parts of hydrogen to make a gas that is then cooled and transported as liquid ammonia. And then my favorite, because, well, I think it's cool, is the atmospheric method, which is lightning. And here you basically see that lightning will break the triple bond of the nitrogen gas and it will combine it with oxygen to form nitrogen oxides. So that's what you see happening here. And then that will dissolve in rain. And ultimately, then plants will be able to use that. And we're going to mention more about this in a little while. So that then brings us to the nitrogen cycle, which you see represented here. And, there's always a lot of different figures and they'll slightly be different. But ultimately, the main concepts are the same, that the nitrogen cycle, the ultimate goal of this is to maintain nitrogen balance in the biosphere. And so what you see here, these are the three methods of nitrogen fixation that we just talked about. And then you see the role that bacteria plays, the very important role of bacteria. And then it moves on to plants that we mentioned a moment ago too. And whereas you have fixation over here on this side, you then see assimilation over here, where you then have the products of fixation getting used toward incorporating them into amino acids. And that's basically showing you, again, the important aspect of metabolism always having a balance to it. You have some breakdown, some synthesis. Okay, now the two primary sources of nitrogen that you are going to see when it comes to the soil part of the nitrogen cycle, they're basically going to be decomposition and also agricultural fertilizers that leach into that soil. Okay, now just remember whenever you hear the nitrogen cycle, ultimately think of how critical it is to organisms and cellular structures, especially since we've mentioned that nitrogen is a critical part of amino acids. So without the nitrogen cycle, we wouldn't have amino acids, and without that, you wouldn't have protein. And so anyone who cares about all of them gains in the gym, you would not have your protein. So nitrogen cycle, kind of important. So now we saw in nitrogen fixation that basically you are going to be breaking the triple bonds of nitrogen, and then you are going to be adding hydrogen to it, and you end up with products like ammonia. And when it comes to this process, next you end up with nitrogen assimilation, able to use that product and incorporate it into amino acids, which I keep emphasizing are critical to us because that is how we build proteins ultimately. So nitrogen assimilation on this slide here, we have three enzymes. The first two, I always laugh when I tell students about them because they're basically self-explanatory. So I can ask you, oh, what does glutamine synthetase do? Well, it synthesizes or produces glutamine. Then glutamate synthase will synthesize or produce glutamate. The third one is the only one that then has a slightly different name to it. It is glutamate dehydrogenase. And this one is basically going to interconvert alpha ketoglutarate and glutamate. So it's going to go back and forth. You notice that the arrow is going both ways. So if you are asked, well, what's going to produce glutamate, it would either be glutamate synthase or glutamate dehydrogenase, whereas glutamine, if I said what's producing that, it's glutamine synthetase. Now, one of the themes you see coming back constantly, I know I sound like a broken record. Whenever we talk about pathways, we then talk about the importance of regulating them. Because again, you need to know when to turn something on and when to turn it off. And so when you think about pathways, I keep mentioning that a lot of times one of the enzymes that you'll want to target is the first enzyme in that pathway. And so if we're targeting assimilation, glutamine synthetase is going to be very valuable to regulate. And here on this slide, we show two ways of regulating glutamine synthetase. The way you can do that are basically techniques that we've mentioned in other pathways over and over. The first one that you see here is covalent modification. Now, previously, when we were regulating enzymes in former chapters, we mentioned a lot of times covalent modification of phosphorylation in terms of regulation. But here, it's not phosphorylation. Instead, you're seeing adenylation. So you have adenylation over here and then deadenylation over here to be able to turn the enzyme into the inactive form versus the active form. Then over here, what we see is the most popular of all regulation mechanisms, which is feedback mechanisms. So especially negative feedback. So if you have a lot of the products, such as the products that are amino acids that are derived from glutamine, for instance, you then have them going back and turning off or inhibiting the enzyme of glutamine synthetase. Okay, so the two ways of regulation, again, are covalent modification. specifically adenylation, and feedback mechanisms, especially negative feedback mechanisms. Okay, so now we're heading more and more into the amino acid side of things. So now we're going to start looking at synthesis of amino acids. Now, what you see here, aminotransferases. These are enzymes that will catalyze the synthesis of amino acids. And sometimes you'll see them named transaminase. Okay, transaminase is the other term for aminotransferases. Now, basically, what you're going to see here is alpha keto acids. And sometimes, for instance, you'll see it here as alpha ketoglutarate. Alpha keto acids are very significant in the synthesis of amino acids because they serve as an acceptor molecule. And you'll notice a lot of these reactions are reversible, but basically what you see happening in transamination uh, is that the NH2 group of one molecule is exchanged with the oxygen group of the other molecule. Okay, so what started out as the amino acid becomes the new alpha keto acid, and what started out as the keto acid has now become the new amino acid. Okay, so looking at it here with specific amino acid over here getting synthesized, we start out with amino acid here. The NH2 group or the NH3 plus group gets swapped with the oxygen over here of the alpha keto acid, in this case alpha ketoglutarate, which is the acceptor molecule. And when you swap these two, notice this original amino acid is now listed as an alpha keto acid, and this alpha ketoglutarate is now glutamate. So this has been a process to synthesize glutamate for us. Now, with everything in biology, remember we have to look at both sides to each story. And so, whenever we mention synthesis, I then want to mention uh, catabolic events, which are the breakdown, the degradation, the opposite of synthesis. And so, when we look at amino acid catabolic reactions, you're going to see the amino acids getting broken down for various purposes. So now, when you break down your dietary proteins and break down the amino acids from them, ultimately the cellular proteins are going to get deaminated, and few things happen. So we look here, deaminated, meaning you're removing this group over here. That will go toward either biosynthetic pathways, so making amino acids, nucleotides, biological amines, or the deaminated, removed nitrogen group will go toward the urea cycle to get excreted as urea. Now, when you deaminate the amino acid and you remove the nitrogen group, you're then left with the carbon skeleton, which is the alpha keto acid part, and that can end up being used by energy conversion pathways. So it can be used with the citrate cycle to produce ATP, it can be used to produce glucose, fatty acids, and ketone bodies. Now, when we're talking about this process of breaking down proteins and deaminating the amino acids and using them for scraps, when we talk about protein degradation, there's actually two different pathways that tend to occur. The way I want you to distinguish them is based on, are they non-selective or the selective pathway? So what I mean by that, the first pathway for degrading a protein is to send it to lysosomes. Now, lysosomes are basically vesicles that are filled with nice, fun, digestive enzymes, so proteases that can digest proteins. Now, this is the non-selective kind of digesting, meaning, any protein that needs to be degraded can be, taken into the lysosome and broken down. There's also the selective pathway, which is the proteasome that relies on ubiquitin tagging. So a ubiquitin little sequence is added to the end of that protein, and that is how the proteasome knows to only grab tagged proteins, only grab specific proteins to pull into that proteasome and break down the protein. Now, when we look at these, another difference I want to mention is the first one is ATP independent. The second one, proteasomes, this highly specific kind of degradation, this one is an ATP dependent process. Okay, so when you think of lysosomes, think not selective. And when you think of proteasomes, think highly selective. It needs that ubiquitin tag. Now, when I say that the proteasome is specific and needs a ubiquitin tag, this figure here, which I know looks scary, is basically a way of showing how you end up with that ubiquitin tag. Basically, the protein targets are covalently tagged on their lysine, so it's usually a lysine residue with ubiquitin, using three ubiquitinating proteins, and you're going to see them here as E1, E2, and E3. And basically, that process will require what's called ubiquitin activation, ubiquitin ligation, and polyubiquitination. Okay, so basically, what's happening is you see here you end up with that ubiquitin tag. You have the enzyme 1 and then enzyme 2 targeting, where you get a transfer of ubiquitin to the enzyme 2. You then see the binding of that complex to enzyme 3, and you end up having ultimately a process where you get so many ubiquitin residues, ubiquitin tags linked together to that target protein. And over here is where you see the target protein basically getting attacked by the complex. And like I said, it's an ATP dependent reaction and ultimately requires a process in which at least four ubiquitin subunits are linked together through the process of having that E1, E2, E3. Now, a little while ago, I mentioned that when you break down proteins and you break down amino acids and you deaminate them, you're removing that ammonia, and if you think about it, that can be a problem inside of your cells and inside your body because that is toxic. So there has to be ways to remove something that is toxic from your body. So basically, what we see here is the urea cycle, so that ammonium produced from that deamination of amino acids can be excreted as urea, or it can be used and recycled to synthesize other nitrogen containing compounds. And when you look at this, I just want to mention that in humans, urea is synthesized in the liver and transported through the blood to the kidneys, where it is concentrated and excreted in urine. Okay, and what's interesting about here and this slide is I always like when you get to see some of the other stuff that we've talked about, and I keep reminding you that one of the big take-home messages in biochemistry is that everything is connected. especially when it comes to metabolism. And so you see kind of some of our former players, such as the citrate cycle, being shown in this representation in terms of looking at urea and then looking at passing urea through passing through the urea cycle to ultimately have excretion. And then we get to one of my favorite namings in science because scientists like to think that they are hilarious, as you can tell by, the way I speak too. I think I am hilarious. Um, and they like cheesy puns. So what we have here it's really titled The Bicycle Pathway. So it's the Krebs bicycle pathway. And it's funny because it is connecting two cycles. So by cycle, two cycles. It's also sometimes called the aspartate arginosuccinate shunt, but that's not as hilarious. So we're going to keep referring to it as the bicycle pathway. And as I just mentioned, the purpose is to connect two cycles, specifically to connect the urea cycle and the citrate cycle. And what you see here is that fumarate produced in the urea cycle. So we have the urea cycle here, and you can have the production of fumarate coming off of that urea cycle to then make aspartate. And so you see this here is the bicycle pathway. You have that fumarate going toward making aspartate in the mitochondrial matrix, and that is linking the urea cycle to the citrate cycle, because you see here you're coming off of the urea cycle, you're going toward one of the intermediates, or I should say two of the intermediates of the citrate cycle, and then cycling back via aspartate into the urea cycle. Okay, so this here is the bicycle pathway, and the reason why you see aspartate arginosuccinate shunt is right here, aspartate and then arginosuccinate over here in the urea cycle. Now, when we talk about the degradation of amino acids, we categorize them into two categories of amino acids. We have the glucogenic and the ketogenic over here. And so when we say glucogenic, it's just like the word suggests. These are amino acids that when you break them down, their carbon skeletons are going to be used to synthesize glucose. Whereas the ketogenic amino acids, when you break them down, the carbon skeleton would be used to produce acetyl-CoA and acetoacetyl-CoA, which both of those, go toward being precursors to the ketone bodies acetone and beta hydroxybutyrate that we've mentioned before. Okay, and so the name of each of these can help you figure out what they're generating when you break them down. And when we look at this figure, the 5 amino acids that are shown in boldface, which you see here, here, over here, those are basically going to be considered both glucogenic and ketogenic at the same time, okay? So any of the ones that are in boldface, they are both glucogenic and ketogenic, meaning that if you break them down, you can use them to produce glucose, which you see over here, or you can use them to produce ketone bodies, which you see over here. Now, it's always important to kind of think of how these things relate to possible health complications or your everyday life. And so in this case, we're going to look at two metabolic diseases that some people end up having. The first one is alkaptonuria, and in these cases, in either one of these, you have the disruption of key enzymes in the ketogenic amino acid degradation pathway. In the case of alkaptonuria, there is a mutation over here in the sorry, I always mispronounce this one, homogentisate 1,2-dioxygenase. And when you have that mutation, you end up having a buildup of this intermediate over here, the homogenetisate. And with this one, if you see that there's a disruption when you're looking at a pathway, if you see there's a disruption in the enzyme right after an intermediate, that means you get a ton of this guy because you can't break it down. So you don't end up with these products and you end up with an accumulation of the intermediate directly before that mutated enzyme. And so what you see ends up happening is this intermediate homogentisate, sorry, I mispronounce it again every time, that builds up and you end up producing a lot of this intermediate over here through oxidation. And you see this in the patient with black color excreted in the urine because the benzoquinone acetate ends up yielding a black color to the urine. Okay. The other metabolic disease from a problem with enzymes in this ketogenic amino acid pathway is phenylketonuria or PKU. And in this one, you see phenylalanine is at the top of this pathway. We are trying to break that down. The enzyme involved in that, phenylalanine hydroxylase, is mutated in people with phenylketonuria. Okay, and so if this is mutated here, then you are going to end up with a major accumulation of phenylalanine because you can't break it down. And so you're not going to get these products. You're going to get an accumulation of phenylalanine and you build up toxic levels of phenylalanine and its metabolites in the brain. And basically, you end up seeing this in neurological symptoms, which can even include severe brain damage. And so when you look at this, if someone has this disease, you're going to want to limit their phenylalanine intake because you know that they're not going to be able to break it down properly. So one of the most important treatments or lifestyles for someone with PKU is to avoid large amounts of phenylalanine in their diet. And please keep in mind that for people with that disease, aspartame or any artificial sweeteners are a big problem for them because that they are big sources of phenylalanine. So people like that, you'll notice there are warning labels on things like diet soda that contain artificial sweeteners sometimes because of that issue. Although my professional opinion, even if you don't have PKU, please avoid artificial sweeteners. They are absolutely horrible for you, even the ones marketed as natural. Just try and avoid things like aspartame, sucralose, stevia, any of that if you can. I highly recommend avoiding that for the sake of your overall health. Now, like I said before, I like when we get to come back to that big take home message of biochemistry that everything's interconnected, especially when it comes to metabolism. All of the different cycles and pathways that we talk about, the different substrates and products and intermediates, all of them end up in this, highly connected network. It's kind of beautiful if you think about it. I know you look at this slide and you're like, that's not what I consider beautiful, but yeah, it's kind of impressive when you think about how everything is so interweaved together. And so this slide is to remind you of all of that connection. And when we look at this, we're looking at amino acid biosynthesis and how they are derived from a lot of the different pathways that we've already talked about in this class. And so when you look at this slide, you see that amino acids are basically derived from 7 metabolic intermediates from 3 of the processes that we've already talked about. So you have glycolysis, you have the pentose phosphate pathway over here, and you have the citrate cycle. Okay, so 7 metabolic intermediates in 3 of these metabolic processes, glycolysis, the pentose phosphate pathway, and citrate cycle, end up being critical for amino acid biosynthesis. And when we talk about amino acids, I want to point out the term essential. And basically, essential amino acids you have to obtain from your diet. And what's interesting is that animals have 10 essential amino acids that we have to obtain from our diet, whereas plants, they don't have any essential amino acids because they can make all 20. And that makes you think, well, we only make 10. We have to get 10 from our diet. Where do we get the other 10? Oh, haha, plants do all the work for us. Like I just said, they make all 20. So we let them sit there, chilling, doing their work, making those 20 amino acids, and then boom, we just eat them up, and yeah, great, problem solved. We get our essential amino acids. Thank you, plants. Now, speaking of plants, that brings us to glyphosate. And glyphosate is a fancy word for Roundup, the herbicide Roundup that you see over here. And so that's the common name of it is Roundup. It's an herbicide that kills plants by blocking tryptophan, tyrosine, and phenylalanine synthesis. It inhibits the important plant enzyme called EPSP synthase. And when you look at this, what's interesting is that if this herbicide targets that plant enzymes so that they can't synthesize those 3 critical amino acids, what we are able to do is there's the bacterial version of that enzyme that's not inhibited by Roundup or glyphosate. And so we can have transgenic plants with this bacterial enzyme, and so they are able to survive Roundup. So basically, what that means is you can have the herbicide kill the plants that you don't want there, meaning the weeds, while your transgenic desired plant or crop survives. Science. So, in this last little bit of the lecture, I just want to go through the biosynthesis of some amino acid derivatives. The first one is something near and dear to our hearts, haha, hearts, heme, which, we've mentioned a few times in this course. And ultimately, it is thanks to the amino acid glycine. So glycine contributes the 4 nitrogen atoms to heme in a series of reactions that occur in the mitochondrial matrix and in the cytosol. Now, heme is not the only molecule that is an amino acid derivative. We also have nucleotides and signaling molecules that are here thanks to amino acids. And so some examples of those are the tyrosine-derived biomolecules. These are also things that we've mentioned previously in this course and that I adore, like dopamine. Who doesn't love dopamine? I mean, come on. And epinephrine and norepinephrine. So these are all three of these, dopamine, epinephrine, and norepinephrine, are all considered catecholamines, and they're critical in metabolic signaling and neurotransmission. We spoke about dopamine in regards to the idea of cocaine and the mechanism of that, because dopamine makes us, energized, happy. It's something that you can really get easily addicted to the reaction of when dopamine is hitting those synapses. And so people end up addicted to things. Even you could be addicted to a person, you could be addicted to porn, to candy, to, various drugs, anything that will activate that pathway. And so what's interesting about that is dopamine, epinephrine, norepinephrine, as you can see in this figure here. So we have dopamine here, norepinephrine over here, epinephrine over here. It's all thanks to the original tyrosine, that amino acid there. They are all ultimately derived from, depending on which enzymes are involved. The last point that I want to make in terms of amino acids being very critical to ultimate biomolecules and visual things that we see happening with bodies and with organisms. Here we have albinos and albinism. And what's interesting is this one is also thanks to lovely tyrosine. And so when it comes to albinos, tyrosine is basically the precursor of melanins, and we have eumelanin and we have pheomelanin. Eumelanin will be the melanin that causes the darker shaded parts of your body. So, for instance, if someone has darker hair, darker skin, they will have eumelanin more active as opposed to someone with lighter shades in hair or in skin, for instance. Pheomelanin, I love this one. Circle star highlight pheomelanin. Any of your reddish pink body parts. So, for instance, nipples, genitals, even the rosiness of your cheeks on your face. Uh, these are areas where pheomelanin is more highly expressed. So I always make the joke that, now that you know biochemistry, you could use that as some smooth pickup lines. Be like, hey, baby, I want to see that nice pheomelanin in terms of the areas that tend to be pink shaded on the body, where pheomelanin is quite a bit higher shaded. So when you look at this pathway, you see that these melanins that give color to your body parts and to your hair and eyes as well, these molecules are made thanks to the ultimate precursor of tyrosine. And so when you end up with mutations in the enzymes encoding for here, what you see, tyrosinase, you end up with a messed up pathway where you don't get the ultimate melanin products. And if you lack these products, you end up with an albino organism that lacks the pigmentation that you would expect to see if there's high melanin production. Okay, so albinos are thanks to an enzyme defect in the tyrosine metabolism pathway. And so that brings us to our review questions. We have a few of them for this lecture. Please make sure to send me each of your answers for these review questions in the Remind app. Another review question here. And please, when you send those review answers to me, please take a picture of the slide so that I know exactly which question you are answering and what answer choice you are making. And that is it for today. As always, thank you and have a beautiful day.